So I would like to thank the organizers for inviting me, and I will say that this title was suggested to me. I don't know which of you came up with it, and I modified it some because I think it's a little early for me to describe exactly how gene therapy will look, but I think that there are some questions that we can begin to answer from the data that we have so far. So I do have to disclose that after many years on the faculty at the University of Pennsylvania, I am now the president and head of research and development at Spark Therapeutics, and our goal is to develop gene therapies for a variety of genetic disorders, including hemophilia. And this is my forward-looking statement. And you can read about all the risks of everything I might say in filings with the Securities and Exchange Commission of the United States. So I will say here that I thought I would try to address several questions. Where are we in terms of investigational gene therapy for hemophilia today? What sorts of people are enrolling in the trials? What does their clinical course look like? And what is the role of the hematologist? And I'm covering this because I hope that many of you in the audience will be investigators on gene therapy trials for hemophilia, and that if all goes well, you will eventually be using it. And I will say this, the pharmacokinetics of it are much easier than what you heard in the previous talk. So I will remind you that gene therapy for hemophilia has been the subject of clinical trials since 1998, and people who are interested in gene therapy have long been attracted to hemophilia because it has a number of advantages as a disease model. It has a very wide therapeutic window. As you know, if you elevate patients' levels to 5%, they'll be better off than if they were at less than 1%, and if they go to 100%, they're still within normal limits. It turns out that clotting factors do not necessarily need to be made in one specific tissue. We know that we make recombinant clotting factors in baby hamster kidney cells and Chinese hamster ovary cells, so we can make biologically active clotting factors in a variety of tissues, and that gives a gene therapist latitude in the choice of the target tissue. We have genetically engineered mice and naturally occurring dog models of hemophilia, and that permits the evaluation of potential gene therapy strategies extensively before clinical trials are begun. And of course, our endpoints are very well established because of many years of history of protein therapeutics. So in the early days of gene therapy, people used a number of different gene delivery vehicles or vectors and a number of different target tissues in that first round of trials that began in 1998. But most of the work eventually coalesced around the use of AAV vectors in the liver. And currently, most of the trials of gene therapy for hemophilia are focused on the use of AAV vectors delivered into the liver through intravenous infusion. The ones in black are for hemophilia B, the ones in blue are for hemophilia A. So there's quite a lot of activity in the space right now, and most of it is centered on AAV vectors. So recombinant AAV vectors are generated, patterned on, and engineered from one of the smallest of all viruses, a parvovirus, which most of us are exposed to during childhood, probably through infections in the respiratory tract. And the vector is generated by deleting the viral coding sequences, which are shown here, and replacing those with the therapeutic gene of interest under the control of an appropriate promoter. So the final recombinant virion is 74% protein by molecular weight, and that is this outer protein shell, and then 26% DNA by molecular weight, and that is the active agent. So what you really have in the recombinant virion is this highly ordered set of proteins on the outside with the active agent on the inside. And for administration in hemophilia, vector is dosed in vector genomes per kilogram of body weight. There was more extensive early work in hemophilia B because the maximum packaging capacity of AAV vectors is about 5 kilobases. So it took some work to make a B domain deleted factor 8 with a very small promoter that could still be efficiently packaged into recombinant virions. This is the trial design for the SPARC Factor 9 trial, and most of the trial designs look very much like this. Right now, they're all for adults, and people have to be screened initially to determine whether they carry a neutralizing antibody to the capsid, whatever it is. The trials have varying approaches to history of Hep C or history of HIV, and then they're typically dose escalation studies. And as I mentioned before, all of these are given now as intravenous infusions, so typically people are coming into some sort of treatment center after the screening protocol is completed, getting their 1-hour infusion, and then fairly intensive laboratory follow-up and occasional visits to the hematologist for the first year, and then into a long-term follow-up study, because all gene therapy protocols now do involve long-term follow-up of individuals. I'll just point out a couple of other things about this initial SPARC Factor 9 trial, because I'm going to use several examples from it. One is that the biodistribution of these vectors after intravenous infusion is overwhelmingly into the liver. These were experiments done in non-human primates to study the biodistribution, but 8 weeks after vector infusion, you can analyze all these other tissues, and you will find that overwhelmingly the vector is in the liver. It does typically have a liver-specific promoter, so even if it is taken up into other tissues, it will not be expressed there because of the tight specificity of the promoter. And secondly, this was a trial that used a high-specific activity transgene, the factor 9 Padua transgene, and that, of course, amplifies the effect of the expression of the protein because factor 9 Padua has about 3 to 8-fold higher specific activity than wild-type factor 9. So first I thought I would just say a little bit about, who are the people who are volunteering for participation in gene therapy trials. And these show the data from the first 11 subjects in this SPARC hemophilia B trial. And you can see that people range in age from 18 years old, just old enough to volunteer for the trial, through people in their 50s. And I would say that we've seen, at least in North America, a representation of adults with severe hemophilia or moderately severe hemophilia participating in the trials. I think that, to me, older adults in many ways are ideal participants because it's a time in life when you might like to get rid of some of the problems on your medical list. And this is one way to do it if things work out well. It's interesting to me that a number of younger people come in, and I actually was spending a lot of time treating hemophilia patients in a time in North America where most patients were on demand. Adults were not on prophylaxis. And so it's surprising to me that younger people come in and say things like, well, I just can't stand taking factor anymore. And these are all people who are on prophylaxis. And, that's always hard for me to grasp that people would feel that way about a medical regimen that makes a profound difference. But the fact is, of course, and all of you know this, that a lot of younger people who have been on prophylaxis for most of their lives can't really remember having had a bleed. And so it's almost as if they're doing a medically demanding regimen without a clear background about why they really need it, not the sense of it that you've had if you were managed for many years on demand. Anyway, so many people with a history of HCV exposure. Most of the trials, I'll mention this later, don't take people with severe liver fibrosis based on HCV, people with a range of joint status, from people who have done very well on prophylaxis and don't have any target joints to people with a lot of target joints, and CRIM positive and CRIM negative for this factor IX trial. This is hard to see, but it's the data that were just published from the BioMarin trial. And again, you see a range of different individuals, although I think the individuals who enrolled in the BioMarin hemophilia A trial tended to be younger than the ones that we had seen on the factor IX trial. And then this is actually the 4 subjects that were presented at ASH on the Spark hemophilia A trial, again, a little bit older. And if we look at comparative demographics here across 4 different studies, we can see that the Unicure hemophilia B trial actually had a median age of 69 in their lowest dose cohort. But again, you see that a good deal of experience with people who have a previous exposure to Hep C, somewhat less experience, a good deal less experience with individuals who are HIV positive, but as long as people have an adequate CD4 count, I don't know of a reason that they could not enroll. So what does gene therapy look like for the participant and for the physician? So, when everything goes well, I'll show you the first case from the Spark hemophilia B trial, 23-year-old man who works as a nurse, and he had severe hemophilia B, CRIM positive, and had been on a prophylaxis regimen with an extended half-life product, but in the year prior to enrolling on the trial had experienced 4 bleeds. Based on his age, of course, he's viral negative. He had done a good job on his prophylaxis. He had no target joints, and he was the father of 2 young children. And this was his course. So on the day of infusion, of vector infusion, we also we bring the patient up to 100% with his clotting factor concentrate. And so then for a while, we can't really measure factor IX levels. But after that, you see that he has a slow rise to his plateau, and that the tracking of his liver enzymes was fairly uneventful. He had a very modest increase in these immediately after the vector infusion. They never got to the upper limit of normal. And so this patient never took steroids. He stopped taking prophylaxis. And 2 years later, this is at his 104-week visit, he's at about the same place that he was when he reached his plateau at about 12 weeks after vector infusion. So this is the sort of canonical ideal course for somebody on a gene therapy trial. So what is the role of the hematologist here? Well, it's looking at who will be eligible, managing patients through the first 40 weeks or so, and then eventually deciding among trials, or we hope eventually deciding among products. So I'll just comment about a couple of other things here. The neutralizing antibodies that have to be screened for. So in that first list of trials that I showed you, these are nearly all different capsids that are being used in these different trials. And studies of populations on 4 different continents, have shown that the frequency of the prevalence of neutralizing antibodies to AAV capsids is similar across continents, and that there's some difference among capsids about the prevalence, but the reality is that the sequences of the capsids are pretty heavily conserved, in the range of 60 to 90%. And so one of the problems that the field is grappling with is what will be strategies for people who present but have neutralizing antibodies to the capsid, and how will we sort through this, and how will we overcome that. So I'll just show you what is it that the capsid antibodies do. This is a case of 2 brothers who presented for enrollment. Obviously, they have the same mutation in their factor IX gene, and they were both maintained on twice-weekly prophylaxis. One of them had had 2 bleeds over the last year, the other none. They're both viral negative, but they did differ in one important respect. The 21-year-old had a low titer 1-to-1 pretreatment antibody, and the 18-year-old had no antibody. So one of the challenges around looking at neutralizing antibodies pre-treatment is that the neutralizing antibody assays are not standardized from one trial to another. And so sometimes people say, oh, well, we should standardize all the neutralizing antibody assays. And it turns out that that's really not easy to do. And the reason for that is that all of these are done by an in vitro transduction of a cell line. And it turns out that the capsids vary pretty extensively in optimized conditions for in vitro transduction of a cell line. So for one, you may need to use a multiplicity of infection of 5,000, and for another one, you may need to use 25,000 to get any kind of reasonable transduction of the cell line in tissue culture. And so it's not so easy to standardize these assays. And in addition to the effect of neutralizing antibodies, some groups have actually described inhibition of transduction by something else in serum that's not an antibody. And so this can, though, have an effect on the outcome. So this is, we could answer this question, but I don't think we need to do it. So what I mean is, what do you think is going to be the effect of these neutralizing antibodies on these two brothers? Do you think that a titer of 1-to-1 is in fact too low to matter? Or do you think that the brother with the low titer neutralizing antibody is actually going to get a lower factor IX level at plateau? And not to keep you in suspense, I'll just show you that the 18-year-old who had no neutralizing antibody leveled out at about 25%, and the 21-year-old who had a low titer neutralizing antibody has a lower plateau level. I think we'll need a lot more experience with low titer neutralizing antibodies and the assays to get a clearer picture of what the effect are. But I do think that, when you see differences in outcome among patients who all got the same dose of a gene therapy vector, the most common explanation is that somebody had a low titer neutralizing antibody. And I mentioned that there are a number of different strategies that people are looking at to overcome these. Right now, what all the trials do is exclude people who have neutralizing antibody titers above a certain level. But eventually, we will want to be able to include these people in studies. And there are a number of different strategies, and you can either manipulate the product or try to condition the patient through plasmapheresis or some other pharmacologic therapy to knock down the antibody titers. So I'm not going to spend a lot of time talking about that, but I do think it's important to be aware of it. The other issue that I mentioned briefly before is that the total experience with individuals who have some degree of liver fibrosis from previous exposure to Hep C is limited at this point, but at least for individuals who don't have severe liver disease, the outcomes seem to be similar to individuals who with no problems. So this is just a 52-year-old man with a history of exposure to Hep C. He had, I think this patient had cleared, it was a so-called natural clearer, but the results are not different for people who have cleared on direct-acting antivirals. And so this person's FibroScan result was not normal, but when he was infused with the vector, he had an outcome really indistinguishable from people who have normal livers. And so I think although there is no experience, to my knowledge, with individuals who on the old Medivir scoring system are an F3 or an F4, individuals who are F1 and F2 seem to have a comparable outcome. Let's see. I'll just mention one other type of case, and this is, where an important role for the hematologist in deciding how to move things forward. But this was an individual who eventually developed a cellular immune response to AAV. And so again, 45-year-old male who treats himself on demand. And after vector infusion, his factor IX level, shown in green there, rose uneventfully. But then he began to show this sort of oscillating pattern, and it was associated, as you see here, with this very subtle rise in his ALT, although not above the upper limit of normal. Eventually, though, his level clearly declared itself as falling, and he was begun on prednisone, and that arrested the fall, and he was left with a circulating factor IX level of around 16%, and he's done well on that. If you looked at these ELI spots that are looking for immune responses to the AAV capsid, they did go up at about 5 weeks after vector infusion, heralding this modest rise in the ALT, and that was really the signal that he should get started on steroids. This phenomenon of an immune response to the capsid has been described in multiple different trials. It was initially described in the first AAV liver trial. It was seen again in Amit Nathwani's trial that was published in 2011. And almost certainly represents a memory T-cell response to the AAV capsid that is reawakened after infusion of vector. Each one of these vectors has a typical pattern for when an immune response occurs. So I always think for phase 1-2, it's better to hold the steroids and see how many people get it and at what time course. And then you can, if a lot of people are getting it, it's a good idea to just go ahead and start patients on steroids prophylactically. But I think it's a good idea first to determine how many people are going to need it. For the hemophilia B trial, so far 2 people out of 11 have needed it. So right now we don't use steroids prophylactically. We add them if they are needed. But again, this is always dose-dependent, capsid-dependent, vector-dependent, and you have to determine in the initial studies whether they're really needed. All right, I'm going to skip through some of these other things and just come to a summary. I think it's early days for gene therapy for hemophilia. I think that if you look at the data from the most recent hemophilia B trials, you can see people have used a range of doses ranging from 5 times 10 to the 11th vector genomes per kilogram in the SPARC trial to 2 times 10 to the 13th in the Unicure trial. Eventually, it will become important what the doses are because there are dose-dependent effects like how long vector is shed in the body fluids. But despite a range of different levels of factor as a result of these different doses, you see that most of them are effective in terms of really reducing the annualized bleeding rate, and that most of the studies have also had very good safety records. Data in hemophilia A are newer, but the paper published by the BioMarin group in December demonstrated a very nice reduction in annualized bleeding rates at the dose that's being carried forward into phase 3. So where we are right now, gene therapy at this point does require active physician management, particularly in the first year, about deciding who's eligible and typically monitoring to initiate steroids and to taper them or to manage them if they are always used. What is the long-term follow-up that's going to be required and at what intervals? Well, for most of the trials, there's a lot of management in the first year, and then typically after that it's reduced to once or twice a year. The federal regulatory agencies vary in what they require for long-term follow-up. In the United States, we are doing liver ultrasounds once every 3 years as part of long-term follow-up, as well as, of course, monitoring factor levels and liver function tests. You all know that there are multiple trials ongoing, ranging from phase 1/2 to phase 3, and that some of the areas of intensive investigation right now include will it be possible to re-administer, and that will require the development of a way to block formation of antibodies to AAV at the time of vector infusion. When will we be able to extend to adolescent populations and eventually to pediatric populations, which is related to this first problem here? When will we extend to inhibitor patients, and how will we overcome pre-existing antibodies to capsid in the population? So I'm going to stop there. I'm happy to take any questions. I'm acknowledging my colleagues at Spark Therapeutics and the investigators in the Spark Hemophilia B and Hemophilia A trials, and thank you for your attention.